Yeah, thank you. Welcome to everyone for today's webinar, which started 6 years ago at the centenary webinar when IUBS turned 100 years. And so if you allow me, I would love to first introduce IUBS very shortly. I apologize, but I can tell you, even if you have joined other webinars, these are new slides because Natalie just constructed and designed them these days. So I go to the beginning just to make clear what the International Union of Biological Sciences is. I should start my laser pointer as well. So as you can see, we are very proud. We are 106 years old. So we were established in 1919. And our goal at that time, and still nowadays, is to represent biology across all disciplines and at all levels, let's say, of the world. Sounds a little bit too large, maybe, but from the international relations to the United Nations and more. And I will tell you a little bit about it. So the idea is to connect bioscientists and to fight for biology. And so it's recognized how important biology is in the year 2025. And so you see here we have more than 110 organizations or national members. And we develop projects, documents, and knowledge in biology and for biology. And this is now a very crowded slide, and I only will read maybe the bigger headings. So we have scientific programs. So you can, if you are, if your country is member, you can apply for support for triennial programs, and there are guidelines in the internet. We have initiatives that can be started in between our general assemblies. We have collaborative projects for biology. And what we are very proud of that we are very, very prominently active on the Conference of Party of the Convention on Biological Diversity. So 19 years back, we started with the first science forum with collaborating partners, and we still do this in the framework of the Conference of Parties. Now the next is the COP17. So we have also educational outreach. We have webinars as today. We support you in sponsoring, and S is missing here. Early career scientists grants can be given if your country is member of IUBS, and we support publications if you don't have other grants to support it. And so maybe what is most important, what are upcoming events, and then I'm already done with my introduction. So you see there is in between these COPs, so COP16 was last year in Colombia. The next one is going to be next year, and in between are these subsidiary body meetings where there is a preparatory discussion what should be the main topic of the COP. And so we are involved in these, have this awful abbreviation, and they are usually called substars. And so we will be engaged in the substar in Panama next month. And then I wish to talk shortly about an early career researchers workshop that is going to take place in context with our General Assembly in Bangalore next year. And before the General Assembly, we have, together with the International Union of Quaternary Sciences, a workshop how paleontological proxies can inform us about climate change impacts on biodiversity. So this is also somehow related naturally to COP and so on and so on. But this gives a nice opportunity to learn from the past for the future. And if you are interested in to join, please, there is an opportunity for early career scientists. And the definition is quite broad. Usually 7 years after PhD, you can apply for this and could join this meeting. And then the last point I wish to make, we will have our next General Assembly. So you see it's counted continuously. Now we are with the 35th International Union of Biological Sciences General Assembly. and that is going to take place after this ECR workshop between 9th and 13th of June next year in Bangalore. And René, in fact, is one of the directors or chairpersons of this General Assembly, and that gives me the opportunity to stop my slides. I hope you got some idea about IUBS, and I give now the word. So welcome everyone once again, and I would love to give the word now to René, because René is going to chair this very nice, I'm sure it will be a very nice and important seminar, webinar today. Thank you very much for your attention.
Thank you so much, Carl-Josef, for introducing IUBS. And of course, today the focus.
Okay. Everybody else is still moving.
Yeah.
All right.
Maybe
we. René's back. Okay.
Oh,
You were frozen, René. So maybe
I was frozen. I'm sorry about that. Unfreeze myself and introduce the speaker for today, Eric Warrant, who really will tell us some very exciting work. Just to briefly introduce him, though he really needs no introduction. And I really want to emphasize the interdisciplinary nature of his work, because he started out as a physicist with a dual degree in physics and entomology from New South Wales, University of New South Wales in Sydney, and then did a PhD on dung beetle superposition eyes, again at the Australian National University in Canberra. And since 1990, he has been in Sweden with did a postdoc with Dan Erik Nielsen at the Vision Group in the University of Lund. And this has, led him to many exciting adventures with deep sea, dim light vision. And on his work in dung beetle eyes, his group was even the recipient of the Ig Nobel Prize. I think he's very proud of that, because it showed how dung beetles can read the stars. And of course, my own connection with Eric has been with nocturnal bees in India, and we've had some lovely times with that. So I don't want to go on more, because Eric has done so much work, written books on, dim light vision, visual ecology, was one of the founding members of the invertebrate vision conferences, which are held once in 4 years, again in Sweden. And, he loves his eyes and his bees, and of course his moths now, and dim light vision and the stars. And that's what Eric is going to talk to us about today. So over to you, Eric.
Thank you very much.
Look forward to your talk.
Thank you very much, René, for that very, very kind introduction. Yeah, I know it's been a pleasure working with you in the past, and being involved in this webinar series. I'm very, very grateful and thankful for the invitation. And I will, without further ado, I'll share my screen here. If I find the right one.
Maybe Simon has few announcements to make just before for
Natalie.
Sorry to interrupt.
Yeah, thank you, Natalie. Just very briefly, hi, I'm Simon. I'm here to help René moderate this webinar. I'm really excited. Also, hi, Eric, Carl-Josef. We haven't seen each other in a while. So I will start a poll just now, just that we see what you actually do. We ask for professional occupation and where you are joining us from. And while you can click through the questions, I will just want to say a few things about how we collect your questions. You can already type them into the Q&A function of Zoom while Eric is still talking. And so we can collect the question already during the talk. If you see a question that you are also interested in, just hit the like button next to that question and we can prioritize the questions then. I hope we can finish by also answering all your questions or giving them to Eric. You may also receive a YouTube link of the recording of this webinar after this session, and you can always find them on the YouTube channel of VBio, but I will send the link. Alrighty, we have 30 people who are actually already answered the questions of 44 that are actually currently present. Maybe we can close the poll just now. So we do have more than a third that are researchers in academia. We do have 2 research managers, 1 educator, a few students, actually bachelor and PhD students, 1 master student and a postdoc fellow, and 3 people clicked other. Nobody's here from industry. We do have a few people from Africa, 43% from Asia, and actually 1 listener from Australia, so from back home. And Eastern Europe is also 1, and Western Europe 11. And I can show you the results also here. And with this, I'm already done and looking forward to your talk, Eric.
Thank you very much, Simon. And welcome, everybody. It's fantastic that there are so many people from such diverse backgrounds and locations that are present. Yeah, so I'm going to, over the next 35, 40 minutes or so, tell you a little bit about the work that we've been doing over the last 10 or 15 years to study a really quite remarkable invertebrate, a moth in this case, that makes an incredible journey in Australia. And before I start, it's just worth saying that it has an incredibly small brain and a very small nervous system and tiny eyes. And despite that, it's incredible what this little creature is actually able to do. So the title of the talk, as you can see, is Incredible Journeys, and it's about how a small moth harnesses the stars and the Earth's magnetic field to make a long journey to a place that it's never actually been to before. And I'll just start by just introducing a little bit about what we understand about the way insects migrate, and that it's probably not necessarily known to very many people, but huge numbers of insects migrate every single day, both day and night. And for instance, at night time, studies have been made that show that literally millions and millions of insects are flying overhead on any given night, especially in the warmer parts of the year, and that these millions and millions of insects can actually convert themselves into tons in terms of biomass. So it's a huge number of creatures that are migrating. But the thing about insects is that almost all of them, with the exception of a very, very tiny fraction, the number of species of which you could basically number on a single hand, nearly all of them migrate from one very broad geographic region to another in search of better conditions. So at the moment here in Europe, for instance, insects are migrating from the north of Europe to the south of Europe, and possibly even into Africa, to escape the coming European winter. And in the springtime, they'll reverse that journey and start moving northwards again. And they're just simply making a giant latitudinal change over a huge distance, but typically from just broadly north to broadly south and in the opposite direction again. That's the way 99.9999% of insects migrate. However, we do know of, as I say, a very tiny number of insects that do something rather different, and 2 in particular that I'm going to tell you about now. And these are insects that begin their journey from an extremely wide geographic area and all travel in different directions to a very, very specific destination, a geographically restricted destination. Probably the most famous of these insects is actually the diurnal monarch butterfly from North America. That is, it migrates during the day, and it starts its journey from a huge area of the northern United States and southern Canada, and flies in different directions depending on where it starts, all of them to in very, very localized region of central Mexico, to a mountainous area that has a specific type of fir tree. And when they arrive there, and they're doing this journey actually in autumn to escape the coming winter, and a huge number of them make this journey, and the journey can be around 4,000 kilometers long from Canada. It's a single individual, and where they're all traveling is, as I say, this very restricted part of central Mexico. And when they arrive, they assemble in trees in their millions in this specific location in Mexico. And here they enter a state of dormancy to spend the winter. It's one single individual butterfly that makes this journey up to 4,000 kilometers, as I said. And they spend the winter in Mexico, and then that single individual begins the migration back again towards northern United States and southern Canada during the following spring. They don't make it all the way, however. It's usually their offspring and their offspring's offspring that completes the journey. We know quite a lot about the way the monarch butterfly is able to make this journey. We know, for instance, that they're able to rely on the sun as a compass for navigation. And the sun is a terrific cue for navigation during the day, because it comes up in roughly the same place every day. It goes down in roughly the same place every day, and it travels across the same track in the sky, roughly the same day in and day out over the space of a number of weeks, while the monarch butterfly is traveling to Mexico. The other migrant that we know of in some detail as well now is the animal I want to tell you about today, and that's the Australian bogong moth, Agrotis infusa. Nowhere near as exciting looking as a monarch butterfly. It's a typical brown moth. I love the look of this moth, I must say. I've gotten very used to its very, very characteristic coloring and patination. It's unlike the monarch butterfly, which makes its migration during the day. This moth, being a moth, of course, makes its migration at night. And the journey it takes is this. I hope you recognize southeastern Australia here. These about 4 billion moths make this journey, and they begin their journey from all around southeastern Australia, from as far away as southeastern Queensland or western Victoria. This is Victoria here in the south. And during the spring, about 4 billion moths travel in different directions from all of these locations and are traveling to a very specific place that they've never been to before. And this place is a small number of cool alpine caves dotted across the ridge tops of our highest mountains in southeastern Australia, in an area which is roughly shown here by my mouse pointer that I'm using here on the map. So it's a very restricted area. And the number of caves we know of is about, currently at least, is about 25 individual caves. When they arrive there, they enter these caves in their millions, and then they line the cave walls like a carpet with up to about 17,000 individual moths per square meter of cave wall. And here they spend the entire summer, which is across the Christmas New Year period in the Southern Hemisphere, about 3 months in a dormant state, which is known as aestivation. And this is a little bit like hibernation, except in the summer. These moths then stay in this state until the beginning of the autumn, when they then leave the caves, the same individuals again, leave the caves, turn around and travel back to the breeding areas where they themselves were born, traveling in these different directions. This is a journey in each direction of about 1,000 kilometers. And on this return journey, when the moths make it back to the breeding grounds, they then mate, lay their eggs and die. And during the following winter, the eggs develop into the larvae and the pupae, and in the following spring, newly emerged adults emerge from the ground to begin this incredible migratory journey afresh. Now, the interesting thing about this is that the parents of these moths have been dead for several months and have never had the opportunity to tell their offspring where they need to go. So all of the information they need for this journey is somehow inside their heads. They just know the direction to travel, and they presumably also know when they have to stop. And this indeed turns out to be the major questions of our research. how is this remarkable migratory feat achieved? How do they know where to go? How do they know the direction to travel in, and how do they know when they've arrived? In other words, how do they know when they've stopped? And so far, we've mostly worked on this first question, but we've even started to get our first results regarding the second question. At the very end of this lecture, I'll tell you what we've found so far concerning the second question, how they know when to stop. So in order to study these questions, the thing we need to do is to capture moths during their migration. And for this purpose, we use a very strong lamp and a sheet strung up between a couple of trees, somewhere on the migratory route of the moth, either the route of the moths coming towards the mountains during the spring or traveling away from the mountains during the autumn. And we can catch a large number of moths with a bright light by doing this. Now, these moths are extremely keen to fly. They're in migratory state. And to do the experiments I'm going to explain to you now, what we do is once we've captured moths, we glue, using contact cement, a metal rod made out of tungsten, which is a non-magnetic material. The reason for this will become important in a moment. It's a kind of a tether, a metal rod tether, which we glue to the back. And if you were to hold that tether between your fingertips, the moth would fly very vigorously on the end of that tether, as you can actually see here in the upper right-hand picture. We then have the possibility to connect that tether to another tungsten rod, which you can just see here in this picture in the upper right, using a piece of small tubing. And the work I'm about to describe to you is work that was done together with my postdoc, David Dreyer, who's shown here on this picture. So once we've done this, we've tethered them, we can then place them into our apparatus. And here is a very simple example of the apparatus that we've used. And here is a film of a moth flying on the end of its tether. That moth is located inside this disc-like arena, which is sitting on a tabletop, in this case under the open sky. That moth is sitting in there and flying, and it can turn in any direction it wishes between 0 and 360 degrees. And with the aid of this little device here, which you see at the very top of the arena, which is known as an optical encoder, we have the capacity to measure the instantaneous flight direction of these moths 5 times every second. So over a period of time, say 5 minutes, we've got the possibility to record over that time period a kind of virtual flight trajectory, if you will, of the moth as it flies during that 5-minute period, because we can track the direction, as I say, 5 times every second of that moth relative to north. And as an example of this, I can show you the kind of virtual flight trajectory that one can record. If we consider this little green dot here as the starting point, then over a period of 5 minutes, a moth could have a trajectory like this. So 3 minutes, 4 minutes, and 5 minutes. And in this particular case, apart from this bit of a loop, the loop before the 1 minute mark, this moth has an extremely directed trajectory in a direction which is just a little bit east of north. And it's very determined to fly in that direction, actually. We can represent that virtual flight trajectory with a vector shown here, this orange vector. And this vector actually gives an indication of not only the direction in which the moth flew, which again is roughly a little bit east of north, but it also gives an impression of how determined the moth was to fly in that direction. So in other words, it gives a measure of its directedness, and that's given by the length of the vector. So we have a direction and we have a length, which describes the direction of the trajectory and how directed the moth was in that direction. Now, not all moths behave like this. Some moths behave like this instead. And this is an example of a moth trajectory where the moth was clearly very undirected, and the average vector for that trajectory is short, and the direction probably not very significant. Now, the reason I'm showing you this is because you're about to see a lot of these vectors in the data I'm showing you that shows you how we answered the questions that we posed in our research. Now, we never only study a single moth. We tend to catch a very large number of moths with our light trap on any given night. So what we do is we study an entire population of moths, possibly many tens of moths on a single night. So we're very interested in what a population of moths do, not just individuals. And so for that reason, we fly many moths in our arena. And we can get a plot like this in the end. So this is again just showing you the kind of data you're about to see. This is an example of the vectors obtained from 10 moth flight trajectories. So there are 10 orange vectors here. Some of them are very short and indicating that some moths were undirected, but many of them are extremely long and highly directed. Now, it's possible for us to account for not only the directions of these vectors, but also the lengths of the vectors to calculate an average vector that represents that population of 10 moths. And that is this red vector that you see here. That red vector then is an indication of the direction that the entire population was flying. In this particular case, that vector is highly significant because it breaks through these levels of significance represented by these circular dotted lines. The longer the vector, the more significant it is. In other words, the more likely it represents that population of moths. So you can see the p-values here getting smaller and smaller as you go out in radius. And the fact that this red vector has broken through the outer circle implies that it represents that population with a p-value of less than 0.001. In other words, with a likelihood of 99.99%. Not all populations of moths look like this, however. Some of them can look like this. And this population is very undirected in comparison. And the average vector calculated from these is very short and has a much, much higher vector p-value, indicating that this average vector is not significant. So this is the kind of data you're about to see. Quite a lot of plots showing this type of data, just so that you're prepared for it. So the first question you might recall is, how do these moths know the direction to fly in? Well, obviously, they've got some ability to inherit this direction via some mechanism, presumably genetic or epigenetic. And so they know the direction to fly in. But as we all know, if we want to head in a particular direction, then we need some kind of a compass. So what kind of compasses might be available for a Bogong moth to use in order to fly in its inherited migratory direction? Well, there are different kinds of cues available at night. The Earth's magnetic field is a very obvious one. We know, for instance, that nocturnally migrating birds rely on the Earth's magnetic field to navigate at night. But visual cues are equally useful. There could be, for instance, information available from the moon and the stars, even temporarily from visual landmarks along the way, which could potentially be recognized. But of all of these cues, the most predictable and stable cues are the Earth's magnetic field and the stars. And the question then for us is to understand whether or not the Bogong moths are actually capable of using these cues. So we can ask first, do Bogong moths use the stars? Our first question that we can investigate. To answer this question, we need a very controlled environment. So this little lab that you can see in front of you here is a lab that's built on land that my family have owned for a long time in Australia, which is located in the mountains south of Canberra, which very conveniently actually is quite close to the mountains where these moths are traveling to in spring, these caves where they're aestivating over the summer. So we've got the possibility of capturing moths arriving in the mountains nearby our place here, and also when they're leaving the mountains. And you can see the little house here on the left, up in the left-hand edge of the picture. That house is where we all stay and eat and plan experiments. And this little lab we built a few years ago to do the experiments I'm about to describe. Now, this lab is made out of completely non-magnetic materials. There isn't a single piece of iron anywhere in this building. And this is very important if you want to study, for instance, the magnetic sense of the moth, because any artificial magnetic cues that we introduce, which are much stronger in terms of their magnetic fields compared to the magnetic field of the Earth, this would destroy our experimental or our ability to do experiments. So this was a very important feature of this lab, that it was non-magnetic. Now, inside that lab, we have two non-magnetic setups. One for electrophysiology, so in other words, for recording from neurons in the brain of the moth in this case, that is completely non-magnetic. And we also have a behavioral setup for studying these virtual flight trajectories I've just shown you. And that's the first setup that you see here. So if you look on the right here, you see that cylindrical arena that you saw in an earlier slide, but this time we've placed a circular lid on the top of that arena. And on top of that, we've placed a piece of translucent paper. In the middle of the lid, you can see the optical encoder. This is during the building of the setup, so you can see the cable hanging down. It's usually not like that. But here is the optical encoder that can record, as I said before, the instantaneous flight directions of moths 5 times every second. So a moth is inside this arena. Now, on the left here, the arena is actually embedded in behind those black curtains inside a set of 3-dimensional Helmholtz coils. Now, these Helmholtz coils allow us to exactly control the Earth's magnetic field in any way we wish. We can change the polarity of the field, we can change the inclination of the field, the strength of the field, we can do anything we wish. But in these experiments I'm about to describe, we actually use the coils to get rid of the field. In other words, the Earth's magnetic field is absent inside the arena. It's a magnetic vacuum. And the reason why we wanted to do that is that we wanted to study the visual sense in isolation without any interference from the moth's magnetic sense, which we've known exists since work we've done a number of years ago, which I come back to in a moment. And here you can see a frame built above the coils and above the arena, which holds a projector. And this allows us, all of this together means that we've created ourselves a little mini planetarium, because we use a program, a free software known as Stellarium, which anybody can download onto their computer or onto their iPhone, absolutely free. And the wonderful thing about this program is that it allows you to project a highly realistic night sky at any location on the surface of the Earth, on any day of the year, and at any time of night. And in the experiments I'm about to show you, we use the projector and Stellarium to project onto this lid the exact same night sky that existed outside the lab on the date and time of the experiment. So this becomes a kind of a mini planetarium, if you like. So the experiment we're going to do now is to place moths in the arena, track their virtual flight trajectory while they experience a natural starry night sky, the same starry night sky that's outside the lab at the time of the experiment. And these are the kinds of night skies that we can produce. We can produce a highly natural night sky in spring and in autumn in its natural so-called 0-degree orientation. And you can see the Milky Way is very prominent, as it is in the Southern Hemisphere. But we've also got the possibility of doing things like rotating the entire night sky by 180 degrees so that north now points south. Another thing we can do is that we can take all of the individual stars of the night sky and spread them randomly across our projection screen in order to produce a starry night sky that's totally random. So it doesn't have a Milky Way and it doesn't have natural constellations. In other words, no possible orientation cues from the sky are present, but the same spectrum of light is available, and so too is the same intensity of light. So these are the stimuli that we presented to our moths, both in spring and in autumn. And here are the results we obtained. Firstly, during spring, this is over 2 spring seasons across 2 years, in a null magnetic field under a natural spring night sky. 70 moths in this particular case, you can see those 70 green vectors and the average red vector for those 70. 70 moths flew in a direction, precisely a direction that we might expect migratory moths to use during spring to travel towards the mountains during that season. So that was very interesting that they could use the natural sky alone without any magnetic field and any other visual cues to fly in that direction. If we turn the night sky by 180 degrees, the moths turn and fly in exactly the opposite direction. Not exactly, but almost exactly the opposite direction. And if we give them a randomized night sky, then those moths are completely random in their orientations. They become totally disoriented. What this shows is, very interestingly, is that the moths seem to be able to use the starry night sky alone to find their inherited migratory direction, the correct direction they should be traveling in during that season. And if that starry night sky is corrupted, as in this bottom plot, they become totally disoriented. If we repeat these experiments during autumn, we see exactly the same thing. But now, of course, the moths are traveling away from the mountains back towards their breeding grounds. And here we see, in this case, 54 moths across 2 autumn migratory seasons across 2 years, traveling in a direction which you might expect them to travel in during spring. During autumn. If we now turn the sky 180 degrees, the moths turn and fly in exactly the opposite direction. And again, if we give them a randomized starry night sky, they're completely disoriented. So this shows that the Bogong moths indeed use the stars as a compass. In other words, they possess a stellar compass, and that they're able to use the starry night sky to causally discern north, south, east, and west, and thus find their correct migratory direction in different seasons. This is only the third group of animals we know of that can do this. In fact, we humans can use the stars in this way, particularly if we use some kind of instrument like a sextant. Night migratory songbirds are known to be able to use the stars in this fashion. And now, for the first time, an invertebrate animal, the Bogong moth, is also known to be able to use the stars like this. There are indeed also neurons in the brain which are responsible for processing these cues, obviously. So we went looking for these neurons by inserting electrodes into the central brain of these moths while we stimulated them with rotating starry night skies. And the part of the brain that we targeted is this region here, which is known as the central complex. This is a very tiny region in the central part of the brain. And this part of the brain is responsible for sensing the world around the moth and using that sensory information to calculate steering commands that allow the moth to turn in different directions. In this particular case, to determine whether or not they're traveling in the right direction and correcting their flight direction if that is not the case. So the central complex is also responsible for generating motor commands, which send signals to the flight muscles in the thorax of the insect, which then change the flapping patterns of the wings and thus change the flight course of the animal. And so this is the obvious part of the brain, which is found, I should point out, in all insects. It's a highly conserved region of the insect brain. And it's, as I say, used for spatial orientation and navigation. And this is the part of the brain we targeted with electrodes to look for cells that might be involved in analyzing stellar compass cues. Just before we leave this slide, I should point out that the compound eyes each have a piece of brain, which they use for processing visual information. This is known as the optic lobe. So visual information is coming through the optic lobe into the central brain. And here is where information about the starry night sky will be arriving from the eyes before it's sent, after processing, to the central brain and the central complex for generating steering commands. So during these experiments, we used the second of our two setups in our little non-magnetic lab. And this is a non-magnetic electrophysiology setup. It looks very similar to the behavioral setup you've just seen. But instead of having an arena inside the Helmholtz coils, we instead have an electrode placed above a moth, which is mounted in a small stand. And so we can use the electrode to penetrate individual visual cells in the central brain, in the central complex of the moth, while at the same time using a projector positioned above the setup to project the starry night sky, again using the free software Stellarium. And during our experiments, what we did is we, and I should say, when I say we, this is actually work of a very talented PhD student, Andrea Adden, in the lab, who was supervised by myself and Stanley Heinze, who you see here. And we had the capacity to rotate the starry night sky above the animal, as I've just said, in different directions. We can also use this randomized starry sky as well and do the same thing. And indeed, we found a large number of cells that responded to these rotations. And here you see a plot of action potential impulses generated in these cells, or in a cell. This is the frequency of action potentials per unit time, or the number of action potentials per unit time, the frequency, as a function of the rotation angle of the starry night sky. And what you can see here for the natural starry night sky is that there is a rotation angle of the starry night sky. In other words, a position of the starry night sky above a moth that would be flying and changing direction, where the frequency of the action potentials reaches maximum. That is not the case if you rotate a random starry sky. They are unable to respond to that in any particular way. It gives an even response. So in other words, we've discovered visual neurons in the central brain that do respond to rotations of the starry night sky. And we found a lot of these cells. We had the possibility also to inject these cells with dye and to actually study their morphology and work out where in the brain they are. I don't have time to show you all of the cells we actually found and recorded from, but suffice to say, we found them in these optic lobe regions of the brain, these parts which are processing visual information. We found them in the central complex of the brain, this navigational center. And we also found them in another region, the output region of the central complex, known as, this region is called the lateral accessory lobes. And this is the part of the brain which generates the steering commands, which are used to change flight course in the flight muscles. And they were found in all of these levels. So the conclusion from that work was that we did indeed find also neurons which code the moth's direction under the starry sky, and that these are also located in the key navigational centers of the brain. So we can add to our conclusion that not only do they use the stars as a compass, but they also seem to have a stellar compass network in the brain for analyzing the stars. The exact nature of that circuitry is yet to be determined, and that's ongoing work. Then, of course, we wanted to understand, now that we understood that they could use the stars as a compass for navigation, another very important thing for us to understand was, how do Bogong moths actually deal with the stars in the real world? Because it's probably well known to most people that because of the rotation of the Earth, the stars appear, in our frame of reference at least, they appear to rotate as the night progresses. So if you take a time-lapse photograph of the night sky over several hours, as was done here, you can see that the stars look like they're rotating. Now, for any animal that uses the stars as a compass, this could be a real problem. The question is this a problem for Bogong moths or not? The other problem, of course, is that not every single night is a lovely clear night where you can see the stars. Some nights are completely overcast. What happens then? And that was another question we wanted to ask. To answer those two questions, we went up behind the lab. So we left our little lab and climbed up the hill behind the lab onto the top of the hill. And we set up two arenas, one of which you can see here at the left, and one of which you can just see, I think, in the distance over here. These are two transparent arenas, which means that the moths that are flown in these arenas have full access to the entire panorama of the world around them. They also have access to the entire dome of the sky. And indeed, they, of course, also can detect and feel the Earth's magnetic field, which goes right through these arenas. This black tent in the middle is where we sat with computers and controlled the apparatus where the moths were flying. This is work done by a really excellent postdoc, Hui Chen, who is currently working on this project. Here is a close-up of that arena. And again, you can see an optical encoder here held by a perspex bar, and the moth would be connected at the end of that rod that you can see there in the middle of the arena. So when we did these experiments, we took one single group of moths and flew them at 2 times during the same night. The number of moths that we eventually recorded from was about 95 individual moths, which we flew early in the evening, about an hour and a half after sunset, which you see at the left here. The sky at that point looked like this. And then about 3 hours later, just before midnight, we flew them all again, after the whole starry night sky had rotated by about 45 degrees. So how did the moths react in those two circumstances? Well, early in the evening, these 95 moths, which were highly directed in this experiment, flew very significantly in a direction which was just east of north, which is given by this red arrow here. 3 hours later, that very same group of 95 moths flew in almost exactly the same direction. There's a bit more spread in the data. They're a little bit more tired after 3 hours of waiting around with a big heavy metal rod on their back. But even after that, even though the sky has rotated by 45 degrees or so, the moth directions here, the average population direction, actually is not significantly different than it was 3 hours earlier in the evening. There is no statistical difference in the directions of those two red vectors. In other words, the rotating stars has had no effect on their ability to travel in their inherited migratory direction, which is roughly northwards in this particular case. How they do this, we're not certain, we're not absolutely sure yet, but as you'll see, we do have a clue. Secondly, under a totally overcast sky, of which we were lucky to have a few of such nights, here we have another group, not the same group, but another group of moths, 44 in this case, and those moths also flew in a direction which is expected at this time of year, which I forgot to say actually was autumn. So this is when they're traveling away from the mountains. Here they're traveling in a direction again, just east of north. And in fact, the direction of that red vector there is no different to the directions of those two vectors statistically. So they're all traveling in more or less the same direction. So in other words, these moths are able to travel in their inherited migratory direction even without the stars at all, which means that the most parsimonious explanation for this is that they must be relying instead, in this circumstance, on the Earth's magnetic field as a compass. So the big question then is, do the moths actually use the Earth's magnetic field for this purpose? We know already that the moths have a magnetic sense. This is work we published already in 2018. But is it possible that they can actually use the Earth's magnetic field to travel in their inherited migratory direction? To answer this question, we went back to our little non-magnetic lab and turned the magnetic coils back on and produced the natural field of the Earth inside our arena, which you saw earlier. This time, however, we used tellurium to create a randomized starry night sky so that they had the full spectrum and intensity of the starry night sky, but no Milky Way, no constellations. In other words, no natural orientation features in the sky that they could potentially use to orient. But now instead, they only have the Earth's magnetic field. In our experiments, we had the possibility of using the coils to turn the entire magnetic field 180 degrees so that magnetic north now faces south. And once again, just as before, we also have the possibility of nulling the field. In other words, removing the Earth's magnetic field entirely. This is again work of Hui Chen. And during spring, when the moths are on their way towards the mountains, moths subjected to this stimulus in a natural magnetic field under a randomized starry sky indeed flew in a direction which would be expected at that time of year. So in this particular case, roughly southwest. This is and if we rotate the starry night sky 180 degrees, if we rotate the Earth's magnetic field by 180 degrees, the moths turn and travel in almost exactly the opposite direction. This implies, therefore, that the moths indeed are able to use the Earth's magnetic field as a compass. This is also reinforced by the fact that if we null the field, just as we saw earlier with the star experiments, if we null the field under a randomized starry sky, they're completely disoriented. The conclusion from these studies, therefore, is that Bogong moths, in addition to a stellar compass, also use the Earth's magnetic field. In other words, Bogong moths use two compasses for robust navigation, a magnetic compass and a stellar compass. So that is the answer, or part of the answer at least, to our first research question, which is how do Bogong moths find their way to their destination 1,000 kilometers away from where they begin, a place they've never been to before. But how do they know when they've reached that destination? In other words, how do they know when they stop? And as I said earlier in the lecture, this particular question is something we've only started to work on now. And it turns out that we have already found a clue to the answer. This is work done by Linnea Roseberry, a relatively new PhD student in the lab. And together with her, we've discovered that the cave itself emits a very strong odor signal, which we've actually determined. It's this compound here. It's an acetate compound. This is emitted from the caves, not from other caves, only their aestivation caves. And we've also determined using behavioral experiments and electrophysiological experiments that spring migratory moths are extremely attracted to this odor. seem to be very interested in this odor as they arrive at the mountains. And this seems to be acting as a kind of navigational beacon that they can follow when they're very, very close to the caves, probably within just a kilometer or two of the destination. This works quite well for an insect like a moth because they have an incredibly sensitive sense of smell, which has evolved, among other things, to sense the sex pheromone of females. So this is this sense of olfaction that they have it seems to be being used to detect even this specialized odor that is being emitted from the caves. We currently don't know the source of this odor, but it's something which is ongoing and that which we are investigating at the moment. But I can say, though, that our behavioral experiments indicate that this moth is completely uninterested in this odor during autumn when they're leaving the caves. The equivalent behavioral experiments which we've done using a tea maze with them flying actively towards either an odor source or a control odor source show that they are highly attracted to the odor source or the odor compound during spring, but they're completely ambivalent to this compound during autumn when they're leaving, which does make sense because, of course, they don't really want to go back in the caves at this point. They want to leave them. So the conclusions of our experiments are that this precision navigation of the Bogong moth from these far-flung areas of around southeastern Australia in different directions relies on many different sensory cues, visual, magnetic, and olfactory cues, the stars and the geomagnetic field as global compasses during the first long-distance phase of the migration, and when the moths are getting very close to their destination during the so-called pinpoint the goal phase of the migration, they apparently are relying on odors emitted from the cave to finally pinpoint their final goal. So this is, as I say, a remarkable ability in an animal that has a brain that quite literally has a volume about 1/10 that of a grain of rice, and a very small nervous system. That's a very impressive feat. And I hope you agree with me that nature is wonderful, and that insects actually possess the most remarkable senses and the most remarkable abilities to solve very complex problems. And with that, I would like to thank you very, very much for attending and for your attention. And of course, before I stop, I would very much like to thank our funders and all of the marvelous postdocs and PhD students and collaborators around the world that have made this research possible. So thank you very much.
Thank you, Eric. That was really marvelous. But I've got so many questions to ask you. But I can see that there are already questions, Simon, in the Q&A.
We do have, I think there's a rather general question, and also the participant introduced the question like that. So maybe perhaps it's a very general question. Does the difference in moonlight has any influence on this perception? Also, how long is the migration time in one direction? How long of an overcast sky will be tolerated?
All very good questions. So the first one was about moonlight. Yeah. We've got no indication so far from all the experiments that we've done that they're interested in moonlight at all. We haven't done a series of definitive experiments where we only investigate moonlight, but so far we haven't seen any effect, for instance, on the movement of the moon. It's possible that they could potentially use the moon as a cue, but compared to the sun, it's a very fickle cue. It's not up every single night, every month. And when it is up, it comes up at different times during the night. It's in a different location in the sky every night, and it has a different size and brightness every night. So it's a very variable cue compared, say, to the sun. So if they do use the moon, then it's going to require quite a lot of calculations in the brain to be able to deal with these variations and a knowledge of how those variations occur across a month. So we don't know the answer to that question yet. It's not impossible, but it's a tough call, let's put it that way, to use the moon. When it comes to the length of the migration, we don't know yet how long it takes them. But we do know from people who have studied insect migration at night using vertical radar, people like Jason Chapman in the United Kingdom, that insects don't necessarily migrate every single night. They're not dumb. They don't migrate on nights where the weather is terrible or where the winds are extremely unfavorable. They like to try and harness favorable winds and will ascend to an altitude where they can find them. So we've noticed in vertical radar records that some nights there's very little migration actually. It's almost as if the moths are waiting on the ground and hanging out until the conditions are better. So it may take weeks actually, but total flight time, it could only take a few days. But those days could be separated by a lot of downtime. But so we don't really know exactly how long it takes. It could take anywhere from 3 excellent days in a row to some weeks with very few excellent days.
Okay, maybe there was something about the overcast sky. How long the overcast sky, the duration of the overcast sky, and might that throw them off? But I guess maybe you should answer that.
Yeah, so far, yeah, our experiments on totally overcast skies where we had very heavy cloud indicated that they weren't terribly thrown off, as I showed you with the data we collected there, and that they could potentially therefore default back onto the other compass, which is the geomagnetic compass. Luckily for Australian insects, we don't have a huge amount of heavily overcast nights. I mean, they do occur, but we have a lot of sunshine and we have a lot of starlight. It's not like in Europe in that sense, but certainly we do have overcast nights, but they are apparently equipped to deal with those nights by having 2 compasses. And I assume, even though most other migratory moths do not travel to a specific location, it's even useful to have compasses to travel northwards or southwards, even if that's a broad latitudinal change in position over a season. And so presumably other insects could have these 2 compasses as well. And in Europe, where there is a lot of cloud, then the magnetic field is probably used much more heavily than it is in Australia.
Okay, before I take a question from what's in the Q&A, I wanted to ask my own question, if I may. You emphasize that moths are reaching areas that they have never been before. So they've never seen these caves, never smelled the smell of the cave. So how do you think this is hardwired? Because you use the term genetic and also epigenetic. So do you want to answer the hardwiredness of
I certainly can, and I'm going to speculate because we don't have any strong evidence for this, obviously. We have done the population genetics of these moths by resequencing the genomes of about 80 individuals from all over southeastern Australia. And we find that they're essentially panmictic, which means there is no population structure, interestingly, except for one single genetic locus, which seems to be highly correlated with the moth's geographic origin. This seems to be a regulatory region rather than a gene, and we don't exactly know yet precisely what it's doing, if it's doing anything. It could be a coincidence, but there is that. But on the whole, it's fairly panmictic. And this has interestingly also been found for monarch butterflies. So my bet is that this is probably more epigenetic. So in other words, these are memories, if you like, that are genetically inherited from the parents and their parents' parents and parents' parents' parents, who knows going back how far. And that when the moth gets close, it has what we call in Swedish an aha experience, which means I've suddenly recognized something that I don't know why I recognize it, but it feels comfortable and I think this is important and that I've come to the right spot. And who knows, the cave odor may not necessarily be the only such cue that they innately recognize. There could be others along the route actually that keep allow them to keep track of their progress, possibly. This is incredibly speculatory, obviously, but we know that genetic memories are real. There are some really excellent studies showing this in not only in insects, but also in vertebrates, that these can be inherited from the parents of individuals that affect their behavior of the offspring. We also know that innately recognized cues are not uncommon in insects, actually. So most pheromones are innately recognized by insects, and foraging insects often have an odor template and a visual template for flowers when they make their first foraging trip, so that they know kind of what to look for. Obviously, those preferences can be changed by learning when the insect finds flowers eventually. But this kind of innate recognition of things is not unheard of. So it's not impossible that this is what's happening with the Bogong moth too.
Okay, I'll just take another question so there were questions about temperature, given climate change and all of that. How might temperature influence, the moth's navigation abilities, and is that worth considering?
Oh, absolutely. I mean, that's a really excellent question. And the Bogong moth, we know, probably does this migration because we haven't published this data, but we do know it has an incredibly low tolerance to temperature. So these moths die when the temperature goes above 39 degrees Celsius for an extended period of time. And this is probably the reason why they migrate, because the areas where they hatch as adults in these breeding areas very rapidly during the late spring easily reach these types of temperatures. So at some point during their evolution, some smart moth has decided we've got to get out of here, and they've found their way to the coolest place in Australia during the summer, which is our highest mountain ridges and caves which are located there. And obviously temperature, therefore, is profoundly problematic for them. So in a world which is getting warmer, we know that the highest temperature for aestivation in these caves is 16 degrees. That is the highest temperature that they will stay aestivating. And beyond that, they will become active and their migratory cycle will be severely affected. At the moment, the cave temperature reaches a maximum of about 10 or 11 degrees. So we're still quite a ways away from that maximum temperature. But what's a worse problem is that out in the breeding areas, with increased climate change and, in Australia's case, longer and more severe droughts, especially during the winter period, where the moths are relying on winter rainfall to create that winter growth that allows the caterpillars to feed during the winter. Already we experienced in 2017 the beginning of a 3-year drought in Australia, which is the worst in Australia's history, which led to these terrible fires that some people may remember in the the end of 2019, beginning of 2020, which were also the worst fires in Australia's history. We experienced a population drop of 99.5% in the Bogong moth population. And these moths are incredibly important for the alpine ecosystem because many endemic and also critically endangered Australian alpine marsupials are relying on them arriving as food in the spring, just as the snow is thawing. They're the first food that arrives. And so during this drought, many of these endangered marsupials perished for lack of food. So this is an incredibly important question, and climate change is a significant threat to the health and future of this moth and all the other animals that depend on it in the alpine ecosystem.
There's a question about lights during the night. Bright lights such as emitted from Parliament House can confuse the bogongs. So how would this, the bright lights confusion be overridden?
Yeah, now that's a good question too. That sounds like it's coming from an Australian listener. So
yes, David Heckham.
Oh, right. Hi, David. No, that's a good question. No, the Bogong moth is famous for invading Canberra, the capital of Australia, every few years. The last time it happened, I think, was in 2013. And They come in huge numbers into Canberra, attracted by the lights of Canberra, and they can clog up the Parliament building, as you've just mentioned, and even shut the Parliament down for short periods, which is considered to be extremely funny in Australia. I find it amusing anyway. But Canberrans on the whole find it a bit annoying because it causes a bit of havoc. But I think the reason for this is not so much light pollution per se. I think it's because unseasonably strong easterly winds have blown them across the mountains off course and above Canberra. And of course, then the lights of Canberra are very attractive. Then they come down in huge numbers and congregate in buildings and begin to do this aestivation behavior. But they normally do that anyway while they're waiting for the snow to melt early in the spring. And then they slowly make their way upwards into the mountains. And in the lower reaches of the mountains, they find lots of cracks and crevices and hidey holes where they congregate on and wait out the thaw higher up, and then they move upwards. Those years they invade Canberra, I think they just use Parliament House instead, because they only stay for a couple of weeks and then they're gone. And we haven't seen a decrease in the number of moths in the mountain aestivation caves during those years. So it's an indication, I think, that they just use Parliament House as a temporary camp. Very convenient.
Okay, well, they're probably even smarter than some of the parliamentarians.
Oh, that's true.
Aha. Okay, maybe then we take yes, there is Dipyamang Ganguly who's asking about, the angles of projection. So, how much does the moth really see, either in your planetarium or in your outdoor experiments? Does the angle or the visual angle
Okay, the visual field. Yeah, no, that's
the
massive.
visual field, how
Yeah,
does that affect?
Yeah, that's one of the advantages of having compound eyes, because the visual fields of compound eyes are truly enormous. They're basically complete wraparound vision, up and down, back and front, except for exactly where the abdomen is. There's a bit of a blind spot there. But compound eyes have a tremendous field of view, which is essential, of course, if you want to look upwards into the sky and see the stars, or see ahead and not crash into things. So that's one of the few major advantages actually of compound eyes compared to eyes like our own. What they can actually see with these eyes, though, is an interesting question. When it comes to the stars are point sources of light. And the only thing that determines how many stars an organism can see is how big its pupils are at night. Our pupil is about 8 millimeters wide, and the pupil of this moth at night is about 10 times smaller. It's about 0.8 millimeters instead. And this makes a huge difference to the number of stars that can be seen. That's why a huge telescope with a front lens, or an optical telescope with a front lens of, say, 3 meters, can see infinitely more stars than we can. We can see some thousands of stars. We don't know how many of the night sky's stars a Bogong moth can see. It could be a few tens of stars. So we don't know if they're able to use the constellations for navigation. The Milky Way, on the other hand, in the southern hemisphere, is a very bright extended stripe of light, which becomes brighter and brighter from the northern half of the sky to the southern half of the sky. And the optics of the Bogong moth's eyes, which is typical actually of nocturnal insects with this superposition type of compound eye, they have a much, much lower F number, as it's referred to in optics, compared to our own eye. So the F number is what you see on the objectives of cameras. The lower that number, the brighter the image the objective of a camera or the eye of an animal creates. So the brighter the image on the retina of an eye, for instance. The Bogong moth sees the extended world around itself 15 times more brightly than we do. So the Milky Way is much brighter for a Bogong moth than it is for us, and it probably easily sees it. And we've got lots and lots of experiments planned now to tease apart these different parts of the sky to see what it can see and what it can't. We intend to find out how many of the brightest stars these moths can see using electrophysiological experiments in the retina, and then create artificial skies using stellarium that contain only those stars, and find out whether they still have the capacity to navigate or not.
Fantastic. So I think we've covered most of the questions except for one, which we will probably end with. And that's about the Earth's magnetic field, flipping over the history of the Earth. So what do you think might happen if, during the course of evolution with these Bogong moths?
That's a very good question. And a very popular question. I get asked that quite a lot, and I don't have a good answer to that. I know we're due for one soonish, whatever that means in terms of geological time, but when that happens, it's hard to predict what will happen. But obviously, lots and lots of animals that rely on the Earth's magnetic field, including Bogong moths, may become terribly lost and may perish. But natural selection being the way natural selection is, currently we've probably got a few oddballs out there that go in the opposite direction to everybody else because they read for whatever reason, they read the magnetic field or the stars in exactly the opposite way than every other does. They will be in a perfect position to deal with the magnetic flip, and it will be their offspring that become the new moth population.
Right. So biology is all about variation, right?
That's right.
So,
Yeah.
and even your maps also indicate that there are outliers.
Oh, absolutely. Yeah. No, as you probably noticed, there's lots of spread in the data, and that could be for a number of reasons. It could be real variation. It could also be the fact they've got these whacking great metal rods sticking out of their back, which, most people wouldn't like, would probably go in all sorts of weird directions if they had one of those stuck on them. And then, of course, we've glued this on. If a tiny bit of glue has obstructed the wing a little bit, and then we've got a slight uneven flight on each side of the moth, then that will, of course, affect the flight direction. We tried to control for that ahead of time, but you never know. There could be some effect nonetheless. There's a lot of reasons. Yeah, and it could be real.
Oh, but still, the data are so remarkable. And Simon, is there any other question you think that?
I think we have a very interesting question still from Nadine, because you told us that, when they go south, they use the odor from the caves to know when to stop. But actually, when they go back north, when do they know how to stop or when?
Right, yeah.
Excellent question. We don't know the answer to that. It could also be an odor specific to that place. But it could be other cues too. They have a magnetic sense, and it could be the magnetic field's characteristics at that location. The magnetic field varies substantially from one place to another on the surface of the Earth. It could be a magnetic signature. But some people have looked at that in monarch butterflies, actually. Have found no evidence for that, to be honest, but it's not impossible.
And one more.
Go ahead, Simon.
A more technical question, again from Dibyaman. I think it was not. He was mentioning also another phenomenon, or does the variance of the vector angle reduce in case of the open area experiments? Because you mentioned, they might just use the Milky Way. So he's asking, is your method in the arena? How well does this, or how is there any interference that you can see then in the field experiments? And also, when both the stellar compass and the magnetic compass are active, does this also reduce the variance?
The variance. That's that's.
Yeah, no, that's a very good question. It does. I mean, if I were to quickly zip back here, I can show you those experiments again. So here, it looks like there's a tremendous variance here, and there is, obviously. But these little red dashes show the 95% confidence limits. And that is actually quite narrow compared to our lab experiments. So here they've got access, as I said before, to the entire panorama around them, to the Earth's magnetic field, and also to the stars. So there is a lot of variation still. And just if you look at the spread of the data in its totality, there isn't much difference actually. But in terms of statistics, at least, the actual variance is at the 95% confidence limits is actually a lot narrower. Because there's a lot of vectors in here that you don't easily see because they're covered by other vectors. So it's much more concentrated than you might imagine. If you represented these just by dots on the outside of this circle, which is a common way of representing this type of orientation data, then they would stack up. You'd see this much more obviously.
Mhm.
Yeah. So the answer is yes, there is actually an indication that when they've got all cues they can possibly have, they are less spread than in the lab.
Okay.
Right, perfect.
Okay, I think we've covered all the questions. Is that right, Simon?
We actually got one more about the neuronal
Oh.
circuits and the brain regions you identified that are specializing in celestial navigation. Can you say a few
Yes.
more words to that?
So sorry, can you repeat that? Sorry, Simon.
Okay, what are the specific neuronal circuits or brain regions identified in the moth that are specialized for celestial navigation?
Yeah, I mean, it's all the same areas that are specialized for all kinds of other navigation, actually, path integration, in insects in general. So these are the different regions and subregions of the central complex of the brain, as well as these lateral accessory lobes, which are the output regions of the central complex of the brain, where the motor signals are being sent to the thorax, as well as there are neurons traveling through the different parts of the optic lobes as well, where they synapse in the lobula and lobular plate, because they're motion sensitive cells often, and they're carrying information into the central complex. So it's all the classic areas which are involved in vision and navigation in insects. Those cells are found in those. Not all cells in all regions, some cells in only one of those regions, others branch from one region to another. So there's all variations actually.
I have a question. Could any of the stars of the Milky Way have particular light spectral features that the moths could be responding to? Particular areas, spectrum.
Yes, this is data I haven't shown. We've used the full spectrum of light in all of these experiments, from UV to red. But in order to investigate the magnetic sense, we've removed blue light from the spectrum because we wanted to investigate whether the mechanism for sensing the Earth's magnetic field is based on cryptochrome molecules, which is one of the two main hypotheses that have been generated to explain how animals sense the magnetic field, which is still a bit of a mystery. And when we remove blue light from the field, from the illumination spectrum, they lost their magnetic orientation. Yeah, so it sort of implies that they probably are using cryptochromes too, actually.
Yeah, but I was talking about, other lights other than blue. Maybe some stars are emitting in particular.
Oh, sorry. Okay, I misunderstood the question. Yes, stars have a much redder spectrum than daylight from the sun. So if you look at the spectrum of a moonlit night and a starlit night without the moon, the moonlit night has a spectrum that looks like sunlight because the moon is actually a reflector of sunlight. Whereas on a moonless starry night, it's a much more redshifted spectrum. So the moths do experience a variable spectrum of light, actually.
Okay, I'm sure you're going to be doing all those experiments in any case in the future.
There's one interesting, not so technical question maybe, but it's also rather a comment maybe. So Melody Green is saying hi, and she was a research assistant for Barry Frost in 2004. And she's so glad to see this work progressing. Have you had the experience of a moth accidentally slipping away and returning to freedom as she was experiencing it with a monarch butterfly? It is a very distinct memory for me and taught me that the good thing about when bad things happen is that it makes it possible for us to remember a moment with much greater clarity than we would have otherwise.
The answer to the question is yes, and how wonderful, Melody, that you've got that connection to Barry, who was like a second dad to me. So I was very sad when he passed away. So it's lovely to have that connection with you. and for those who were in the audience that don't know, Barry Frost was the person together with Henrik Mauritsen that pioneered the apparatus that we use in our experiments, this flight arena for use on monarch butterflies. Anyway, yeah, they very frequently break off the tethers and fly away. And we tend to actually release them at the end of the experiment anyway. We usually it's easy just to remove this little stalk and let them go. Because it's a waxy cuticle that it's attached to, and it's not extremely tightly attached. So we tend to just remove the stalk and let them fly away.
All right. Yeah, she says thanks.
Thank you.
And maybe one last question.
Okay, go ahead.
Divyaman has one more. If he's not wrong, dung beetles, Scarabaeus satyrus, also take cues from the direction of the Milky Way.
That's true. Absolutely. I was very heavily involved in those studies. That's true. Yeah, that is in fact the first insect, or actually, yeah, the first insect that is known to be able to use the starry night sky as a compass for short distance orientation. It's a slightly different thing than the Bogong moth, because the dung beetle, its main job in life is to roll a ball of dung in a straight line away from the furious competition of a pile of dung, where there are lots of other beetles doing the same thing and will happily steal your ball from you that you've made if you're not careful. So you've got to leave there and get away in a straight line and never, ever, ever roll back into it, because then you'll certainly lose your ball to a big, tough thief. So you've got to go in a straight line. It doesn't matter what direction, any direction will do. And it goes, and you have to roll for about 10 minutes to get, some tens of meters away from the dung pile. At that point, you then dig your dung ball down into the ground and lay your egg in it. That's the idea. Now, because it's a random direction, they only use the starry night sky as an orientation cue to travel in any random direction. There is, in fact, no goal here. The goal is to get away from the dung pile. It's not to go somewhere. And the Bogong moth is completely the opposite. So the Bogong moth is actually going somewhere. It's got a goal far away that it's never been to. So it's using the stars in a very different way. It's using it as a true compass, actually, to discern a direction relative to north. The circuitry need not be all that different than the dung beetle circuitry in the central brain, but the purpose of the stellar compass is very different.
Okay, perfect.
Right. I think we'll take then the last question, which was by Gillian Bowser, who's asking about migration in the butterfly, the painted lady, Vanessa Carduai, where there are multiple generations that, make this long migration. So she was just asking a curiosity question as to, whether you can speculate how multiple generations, can, I guess, maybe similar to the monarch butterfly, but maybe you want to take that question.
Yeah, it probably is very similar to the monarch, I think, because they do it in several generations. So it's a couple of generations that completes the return journey in the monarch. And the same is true in Vanessa, in the painted lady. And presumably the offspring also have an innate sense of where they need to travel, so that they are able to fly in the right direction. And they are almost certainly using the sun as a compass, I would say. In fact, I think there may have been a study on that, in fact, showing that was the case. So they're probably very capable navigators as well. That they do it in multiple generations, of course. They do a very long migration, I should point out. It's similar to that of the monarch. They, in contrast, don't go to a specific destination. They're a general migrant.
Okay. I think, my gosh, I think we've had a wonderful session, a wonderful evening for me in India and for mornings in other parts of the world, or late nights, or wherever you are. I hope when you look up at the starry sky, assuming you can see it, in Bangalore we can't with the light pollution. So I wish, thank you very much, Eric, on behalf of IUBS, and of course, Carl Josef perhaps can say the last words, thanking on behalf of IUBS. Carl Josef, please.
Okay, thank you. You could have done it perfectly well. Thank you, Eric, and thanks all participants for the amazing discussion. I also would have had a question about evolution, but you answered it a little bit. So it's really amazing. One can think a lot about this system, which is really amazing, how biology evolved and what it created somehow. So it's really good. Thank you very much to all of you, Eric first, and all listeners, and René, Natalie, Simon.
Thank you very much for having me. It was a pleasure to be here.
Yeah, thank you, and bye-bye to us, to everybody all over. Bye-bye. Bye.
Thank you. Bye-bye. Bye.
Thanks, Eric. Bye-bye.
My pleasure.
Yeah, bye.